All right, welcome to Neerja's, it's not thesis defense, thesis talk. I think you can see by all the people here and online, very diverse group of people that Neerja's has a lot of connections around this world and around Berkeley. And that's been a benefit to our lab. So whenever I do this talk, I always go and look up the first emails I had with a student. And yours were not embarrassing at all. The only thing embarrassing is that we did it on Skype, so. But yeah, Neerja's been a fantastic group member. I think everyone in our group can attest to that. She's really helpful in every way, with keeping the lab running and just like the best lab citizen I could imagine. And she's also done great research. You're going to hear about her research project, which I think is pushing towards like biological microscopy with hyperspectral, which is an emerging topic that I think is important. And she's figured out ways to make it practical and demonstrate it in the real world and for like real people who are doing real science with her stuff. And she even installed one of these microscopes at Stanford, which is a big like boon to the impact of the technique that people can now just use it. I also will say, like, over these years, I've had so many of my colleagues come up and say, oh, Neerja helped out with this mentoring, outreach, etc. It's department service thing. She's so great. And I'm like, yeah, of course. And she recently won an award for that. That was actually spearheaded by Kurt Koitzer, who was so impressed by the impact she had on mentoring some more junior students. And I think that is a big lasting legacy she's going to leave, not just for our group, but for the department and the whole like scientific community beyond that. So like that's a lot to be proud of, even if it doesn't show up on your CV in some ways. So happy to hear your talk, and we've been very lucky to have you in this group.
Okay, thanks, Laura. All right, hi everyone. We're gonna get started. Okay, I think we have a little bit of time for questions in between, so I'll break it down into different sections and then I'll pause when we have time for questions. So today I'm going to talk to you about my thesis work, which has been focused on computational hyperspectral microscopy for bioimaging. So let's take a step back, and just so we're on the same page, light is made up of electromagnetic waves. This means there's oscillating fields of electric and magnetic energy that propagates through space. And just like sound is made up of different pitches and different notes, light also has different wavelengths or different frequencies. And when we see color, we're actually detecting light that's made up of these different frequencies. So for example, you have ultraviolet light, which is on one end of the spectrum, and then we have red and near infrared on the other end. And this spectrum extends even farther. So when you go and get an X-ray, that's even shorter wavelength light than we can see in the visible. And when you're listening to the radio, that's even longer wavelength light than what's shown on here. So light touches us every day and we interact with it all the time. And not just us, but all of the matter around us. And this to me was really fascinating and kind of the first spark towards my PhD. I love thinking about light matter interactions. I remember I wrote my college essay about how I see light bouncing off of photons, and it's interacting in different ways. So for example, light slows down and it bends as it passes through water. It also gets absorbed and then it reflects as it passes through like the surface around us. And that's how we see color of objects. And then lastly, as we start looking at molecules and atoms, light interacts with the electronic structure and the molecular vibrations in these materials. And that helps us understand what are the elements that this material is made up of. And that field is called spectroscopy. So my previous work was in the field of spectroscopy. And when I came to Berkeley, I wanted to extend it to 2D to focus on imaging. So my goal was thinking about, could we capture all of this rich information around us? And I'm imagining a world where we have these like tiny microscopes or sensors kind of everywhere to capture this rich information. And I feel like now is a ripe time because with the advances in computation, we have all of these tools for being able to deduce and make sense of this information, but we need a way of capture it. And I use the term information quite loosely. There's work in our lab being done that's more strictly along information theory. So we can think of this as more just like content. How do we try to build high throughput, high content systems to capture all of this biology. The other last thing that's exciting about light is it's great for looking at things like cells and tissues. And that's because it's a non-destructive method to be able to measure, things that are happening at the cellular level. And so we're able to probe things inside cells without destroying them and being able to watch them live. In my research, I focused on hyperspectral imaging. So what that means is we're measuring the electromagnetic spectrum at every point in the scene. So that spectrum I showed earlier that was going from ultraviolet to near infrared, instead of just measuring rough quantities of red, green, and blue, we're actually trying to get the exact intensity information at every wavelength. And that's important because that will tell us those material properties. What we capture is this hypercube that has X and Y spatial axes, and then the third axis is wavelength. And there are many applications for hyperspectral imaging. We start off from microscopic applications like looking at cells and tissues and small materials and nanostructures, but this extends all the way up through satellite imagery when we're looking at the planet Earth and we're seeing all of the landscapes and a lot of the agricultural applications. So my work focused more on microscopy, which is on this side of the spectrum. And the main tool that our lab uses is this field called computational imaging. And what that means is that when we make a picture, we need to get all of the light from a point on the object to land on a camera. And when we do traditional imaging, the way we do that is we use a lens that focuses the light and redirects the rays from one point on the object to one point on the sensor. However, when we do computational imaging, we do something a little different. Instead of needing all of that light to land on one point in the sensor, we actually encode it using some kind of optics. So it creates maybe a picture on a sensor that a human cannot read, which means then we need a machine or a computer to decode that picture into something that makes sense. And that field is called computational imaging, where you have the co-design of both the optics in the system and then also computation or algorithms that work together to try to capture this rich information from our object and make sense of it. And the benefit is that computational imaging can potentially help us make systems that are more compact and higher spectral resolution. And that's what my PhD focused on. So I'll talk about some projects that do this. And this work really builds on previous work that's been done in this field. So for example, a lot of work has been done in photography, and my work is building on that by focusing on microscopy. Many scientists have developed different kinds of computational hyperspectral imaging systems. And so we're inspired by them to develop new architectures. And then on the reconstruction side, we are doing scientific imaging. So we're taking learnings from prior work, but then also being careful about what are the applications that we're targeting? What are the assumptions that we can make about our object, because we don't want to hallucinate or give wrong information. And then lastly, as some of you know, I came from industry and then I went into my PhD. So I always feel inspired by making actual technology that impacts people. And so the hope is to turn some of this work into products or commercial systems that can actually reach people. And the way we can do that is by making it compact and making it accessible. So now there are many different ways to capture hyperspectral data. The first is, you can imagine you have that data cube and we capture one spot at a time, and we measure the full spectrum at every spot. So that's what the first project is going to do. The second project is going to focus on snapshot techniques, which means we capture that entire data cube at once. But you'll notice that then we potentially sacrifice resolution, which means like how small are the features that we can capture. And we'll talk about a way to use computational imaging to mitigate that. And then the last project has to do with spatial frequency scanning. So this is kind of like a new technique where I started working in this field, and I thought, well, we haven't really thought about capturing this information in this new way. And our lab actually has prior experience in a technique called Fourier tychography, which might be suitable for this. So we'll learn about a new way of capturing hyperspectral imaging. Okay, so we'll get started with our first project, which is the diffuser spectrometer for optical coherence tomography. This work was done in collaboration with Joe Malone, who's at Vanderbilt, and also Eric Markley, who has done almost all of my projects with me, and it's been a joy. So let's get started. So in this project, what we're doing is we have a very simple optical setup, which is a diffuser and then a sensor. And we capture something called a speckle measurement, which I'll go over. And then we take that speckle measurement, which is encoding the color information from the light, and then we put it through an algorithm to get back what is that spectrum. So on the right, you see that wavelength versus intensity. So that's telling you what is the component of every color that's made up of this light. And the whole goal of this project was we had a collaborator who was really excited about making OCT mobile. So OCT is this eye imaging technique where we shine light into a patient's retina, and we can actually see a few millimeters or a few like 100 microns deep into the tissue and get structural information. And the goal is we want to make this compact so you can use it in the field. And it also has applications for cell imaging. The way traditional spectrometers work, so spectrometers are these devices that are used to measure the colors of light, is that they have something called a grating. You can imagine that Pink Floyd picture where you have that triangle and then it separates light into different colors. Same idea, different technique. And then we take those colors that have now been separated and then we focus them onto a sensor using a lens. So that way we get one spot for every color. And this makes a pretty bulky device because the resolution is related to the size of this device. So one way to make this smaller is that we get rid of that focusing lens, but that means we need a way of encoding that information on the sensor so we can unscramble it later. And so to do that, we use something called a diffuser, which essentially is going to be a bumpy piece of plastic that's able to scatter this light and create that speckle pattern. Then we run something called an inverse problem. So we solve for what our input was given a matrix that represents our system and then given a measurement. And there's been work that's done this in the past, and our goal is to make this more accessible. So we decided to use something very simple. We used Scotch tape. So this is like the tape that you have in your office. You can do spectroscopy with it, turns out. All you need is a way to make sure the light that's hitting the tape is collimated. So you use something called like maybe a collimating lens, and then you need a pretty sensitive camera to capture it. And what happens is as that light passes through that bumpy piece of plastic or tape, it forms these tiny speckles. And that's because of interference within the light with itself. And so what you get is this speckle pattern on an image on a sensor. And this speckle pattern is unique for every wavelength. So as we change the color of light, you see that the speckle pattern is stretching or changing. So we create a calibration matrix where we measure the pattern at every single color and we stack that up. And then we know that what we are going to measure is going to be some weighted sum of these speckle patterns, and then with a little bit of noise. So then we solve this very simple inverse problem, and we can actually solve it using like Tikhonov regularization or pseudo inverse, so kind of simple linear algebra techniques to get back the input spectrum. So now let's look at some results. We were able to demonstrate that we can get roughly like 2 nanometer resolution with a device that's about like 5 to 7 centimeters small. And we were able to reconstruct broadband spectrum. So this is light from different sources of LEDs that we were able to match using, again, the Scotch tape diffuser. And we were lucky that we were able to publish this work in Optics Letters, and I was able to present it at Photonics West. So really quick, the applications for this was optical coherence tomography. That's still our end goal. And now we are actually continuing this work. I know Taeyong in the lab is looking at stacking multiple layers of diffusers or optics to be able to get even better resolution in a smaller form factor. Okay, I think we have time for one question. So does anybody have a question? Otherwise, we'll move on.
That's what I'm doing.
Okay. All right, if there are no more questions, we're going to move on to the next project, which is even more exciting. Okay, so this next project took up the bulk of my PhD, and it's in very strong collaboration with the one and only Eric Markley, who has, again, been such a joy to work with. Okay, so in this project, what we did is we built this small attachment for a traditional benchtop microscope that turns it into a hyperspectral microscope. This hyperspectral microscope captures a 2D image. So we had talked about that 3D data cube. We actually squash it down into a 2D image that kind of looks like nothing on a sensor, so you can't really decode it yourself. And then we run a reconstruction algorithm to get back the scene. So in this case, we are able to build up a 3D data cube imaging these cells that are stained with fluorescent dyes. And our device is very small, so you can see here it's only 6 inches and it can actually be attached to any benchtop microscope. The applications for this work is anything from like cell imaging to bioassay beads. We're really good at looking at tiny dots. So there are new applications coming out like protein sequencing, which I think could benefit from this work. And it's built on a lot of previous work in our lab. So in my first year, I joined the lab all excited and gung-ho, and a lot of research in science is about timing. So I was very lucky that I joined the lab right when this photography work was coming out. It was led by Christina Monakova and Kyrilos Yani in our lab, and they were kind enough to let me get started on this project, and I helped a little bit with the calibration and then ended up being on that paper. So that was very exciting. So in that work, what they were doing is we were using a diffuser, so similar to the diffuser we saw before, and then we have a special type of camera which has a spectral filter array on it, and that spectral filter now is what's helping us understand which colors the light is made up of. So this is a little different. We're not relying on the diffuser to scatter the light into different colors. We're actually using the spectral filter to get the color information. And the way it works is that the spectral filter is kind of like a RGB Bayer filter on a camera, but instead of 2x2, it's 8x8. And you have a specific color that passes through every subfilter. So by using the spectral filter, we're able to get the full UV visible near infrared spectrum for every point in our scene. But you can imagine now we've made this filter bigger, so we need every point in our scene to land on every filter to be able to measure its color information. Otherwise, we don't have information about the other colors. So to do that, what we do is we use this idea of multiplexing, where we spread out the light from every point in our scene onto our sensor using the diffuser as before. And the pattern that this diffuser is making is this sharp caustic pattern. So this really sharp point spread function that if the point moves a little bit, this whole pattern shifts, and we can detect that change really easily using our algorithm. So we built this experimental setup in our lab, and it's actually quite simple. It's only 3 components. So you have something called a relay lens, and what that does is it takes the light from inside the microscope, puts it outside, and has collimated light hitting the diffuser. So we talked about before how we had that fiber and then a collimating lens. That's kind of what this relay lens is doing. And then we have our diffuser, which is now shaping the point spread function. And then we have our spectral camera, which we actually made in our lab. So we were lucky to have a collaborator that could give us our spectral filter, and then we just glue it on to a regular monochrome camera. And this was through a lot of iterations. So when I'm showing, the nice thing about a research talk is you get to see all the pretty results, but there was a lot of intermediary work in between. So over the course of like 2-3 years, we actually redesigned the setup, and now it's in our collaborators lab down at Stanford. The optics are a lot more professional. There's like an off-the-shelf bill of materials you can use, and we wrote instructions for how people can build this themselves. And we also made a code repo where they can run the reconstruction algorithms themselves. So now let's talk about the computational side. We talked about the optics, but the computational side is just as important. I won't go into the specifics, so happy to talk about this in Q&A, but for every imaging system, what we need to do is we need to model what's happening to the light. So for example, we start off with our object, which is this Cal data cube. We need some way of getting from our object to what our measurement looks like, which is B, this black and white picture. And then what we do is we subtract our estimate of what we think our measurement looks like, which is this first term with the summation, from what our actual picture is. And the hope is if you match those 2 terms, your estimated picture with your actual picture, then whatever you thought your object was is probably correct. It's a little bit like, you know, when you play Guess Who, you ask a lot of questions, and the goal is when you look at all of the yeses, those match what your partner gave you, and then you kind of know who the person was that they have in their card. So you're kind of back solving this problem. We also incorporate regularizers or priors. So these are basically terms that give you additional information about what you think your object was. And the reason this is important is you remember we had that 3D data cube, which had spatial along 2 axes and then wavelength along the 3rd. And we need a way to squash that down into a 2D measurement. So we're trying to solve for more unknowns than we have information for, and that's where the priors come in. So in summary, we're going to reconstruct this 3D data cube. So what we do in practice is we take a picture using our microscope that looks like this. And then we also feed in some calibration data we had collected earlier into our algorithm. And what it gives out is that recovered spectral object. So what I'm showing here is simulation data for this system. Now let's talk a little bit about this reconstruction approach. You'll notice that we're doing a physics-based reconstruction where we're incorporating information about what our actual imaging system is doing. And the other end of this is if you had an imaging system that you didn't know anything about, but you could capture a lot of data, then you could use a more deep learning oriented approach. And that's kind of another direction the field is taking right now. And both approaches involve incorporating priors, which means you're kind of biasing or you're incorporating prior information. In the deep learning approach, it's dependent on your data set. In our physics-based approach, it's dependent on those exact terms. And then my opinion is that with scientific imaging, you could potentially do more. I personally prefer white box approaches because then you know more about what's happening to your system, but I think there are ways to do both. And then this is kind of good to know because nothing works. That's what you learn about in research and science is nothing works. Every day you go to work and you're like, oh, today nothing worked. It's a great day. And that's kind of the case for most of the PhD. And so you go in and you keep trying and you keep trying. And as like Eric and I spent a lot of time debugging the optics together, debugging the reconstruction algorithm together. It's a lot more fun to sit in the dark when you have somebody else next to you, I have learned. And there were a lot of issues, like our system destroys a lot of light because we have these narrowband filters. We have reconstruction problems because we didn't do our calibration right. We were getting these weird artifacts. So it took a lot of iterating to actually get this system to work. And I think this is useful, which is how do we think about a computational imaging system and how do we go about debugging it? Because as engineers, the one thing we learn is debugging. So there's a lot of different stages. There's stuff on the biology sample prep side we could fix, and we did fix. There was hardware improvements that we made and acquisition and calibration improvements. And then there were also computational improvements that we had to make to be able to get everything to work together. And so the point is like, there's no one fix. You really have to understand your full system and be able to deep dive and fix any part of it to be able to get a system functional. So that's kind of a good practice in engineering in general, is know your systems. Okay, now we get to move on to results, which is the exciting part. So first thing we measured are these fluorescent beads. So these are just like polystyrene or plastic beads. They're really good mimics for cells because they're small and round and they're really bright. And we were able to see these. And the exciting part is that we're able to image these beads smaller than the resolution of the superpixel. So by that, I mean, we saw that there was that 8x8 tile before, and typically you would need each point to land on the entire superpixel, which is shown on the top left. But we're able to resolve features that are smaller than that. That means we're able to beat the traditional limits using computational imaging. And then if we look at the spectra for the components in this scene, it matches the ground truth, which means our spectral estimate is also correct. Sure. Really quick. Okay, now you can see these. We'll turn the lights on after we're done with the results. So please remind me. Okay, but this is a snapshot technique. That's like the main advantage of it, right? Where we can capture that whole data cube at once. So then we tried imaging a moving scene. So these are the same fluorescent beads as before, but now they're moving because they're drying in a water droplet. And on the right, you can see the reconstruction where some of the components are moving and we're able to capture a time lapse using this system by taking multiple acquisitions. And then we were able to image fixed fluorescent cells again. Yes. Yeah. Sure. So over here, some of these beads are not resolvable because they're closer than the resolution limit of even our diffuser. So that part is expected. And then in other parts of the scene, you can resolve the beads that are at least the diffuser's autocorrelation width apart. So I would say it's, and then the artifact depends on how much total variation. So basically that's how much smoothness we enforce into our image. And that's another thing about computational imaging is that you have to tune all of these hyperparameters. So it's a little bit customized to that sample. So what we did is we first looked at the static case, make sure that we got that working right. And then the dynamic cases, those same parameters applied to multiple scenes. Okay, Mickey's special, so he gets to ask a question in between. Okay, and then we looked at fixed fluorescent cells. So for this, we looked at these human Huvic cells. So they're basically like, think of them as like round and flat, and they're a standard cell line that we can get from our lab. So this work was done with our collaborator in Lydia Sohn's lab, Stephanie Eberly, who was awesome. And we stained these cells with different fluorescent dyes, and we're able to get back the cells. And so on that note, you can see like here, we're able to resolve two cells that are right next to each other, but they're closer than that superpixel limit. And we're able to get the spectra back. And then this result took us a really long time to be able to get. So our goal was, oh, it'd be so pretty to see a picture that spells out Cal, because go Bears. We also tried a bear. The bear did not work, but we got Cal. And they have this amazing technique where they can actually pattern cells onto a slide in like specific places. So we custom designed this slide for us, did a lot of iterations, and we're able to get like rough imaging working right now for this sample. So on the left is the measurement, in the middle is the reconstruction, and then on the right is the ground truth. And this is the spectra where we can see that the spectra matches the ground truth. And just for comparison, this is what we got before we fixed everything. So things don't work for a long time. And then lastly, I showed the results down at Stanford. So this is these are results off of that setup at Stanford, where we imaged these special beads that they have for doing bioassays. Basically, that means they look at different ways to like conjugate proteins with other proteins, and they have these beads that are spectrally coded. And these beads have very narrow spectra. And so you need this technique because you want to be able to resolve beads that have just a slight variation in spectra. And so we could see these beads, and with our technique, the spectral reconstructions match the ground truth. Okay, so that was all kind of the main results. The other fun part about this project is I was able to mentor a lot of students while working on this. So Yesho was a senior and master's student that worked with me for 2 years, and his focus was trying to make this system portable. We made a benchtop system, but the idea is this can also be used in the field. And so you remove everything that's the microscope part, and you just use this imager, and the goal is to make it small and portable. So he looked into this idea, and he was able to make some advances and also present his work at Photonics West in 2023. And then most recently, like last semester, Nellie and I mentored a group of students in Laura's comp imaging class, where they tried to use Gaussian parameterization to represent this hyperspectral object. So happy to answer questions about this later, but the cool thing is like, this is their reconstruction algorithm running in real time, where it's guessing what the object should be. So it's matching the measurement to what they actually got, and they were able to get it almost looking like a C. This is all work in simulation, but they did a great job. And then I also mentored some other students, for Preeti and Ethan, just for a semester, and they did some great work on this too. So in summary, I did this project. There's my tiny floating head above the podium that focus on microscopy, because they have big podiums, and I got to present this work like at three times at that conference. So I got to go to Portugal, and then I got to go to Italy, which was really fun. And then we're working on a manuscript for this work right now, and that's still in the works, and we're getting close to submission. So any questions about this project? Let me turn up any questions on Zoom? Okay, great. We get to go on to the third project, which is an idea I had in my first year of grad school. And then I actually pursued it in my last year of grad school, because all of the other projects took a long time. So in this project, what we're aiming for is even more resolution than we got before. So again, resolution is how small are the objects or features that we can resolve. Can we turn the lights back up? Thanks, Regi. What are the features that we can resolve using this system? So a bit of background, our lab has developed this technique called Fourier tychography microscopy. And what it does is it captures really wide fields of view and really high resolution. Let me see if I can turn the brightness a little down. Oh, I know. All right, that's good enough. And so it captures really wide fields of view and really high resolution. And the way it does that is it captures multiple acquisitions using different angles of light hitting your sample. So we have a microscope that has an LED array. And using that LED array, we shine light from different angles. It passes through our sample. We have a normal microscope on the other side. And so we have our camera that takes a picture. We take a bunch of low resolution pictures, and then we stitch them together using a reconstruction algorithm into a high resolution image. And so our goal is to do this at multiple wavelengths because we want to do hyperspectral imaging. And the reason I'm interested in this is because there's been some new work where we are seeing that the way light passes and absorbs or transmits through tissue samples might be able to be a way to do label-free digital pathology. And that's really exciting to me. And what's new about this is so far we talked about ways of doing hyperspectral data capture by scanning through our data cube, either in space, or people have also done this through wavelength in the field. And then we also talked about a way of capturing the whole thing at once, but at low resolution. A little bit better with the diffuser, but still not great. Now I'm thinking, what if we could scan in something called spatial frequency? So again, this is like music is made up of different notes. You can also look at a picture and it's kind of made up of light at different, it's made up of patterns at different frequencies. And so if we scan and capture those different frequencies, then we can go to really, really high frequencies. And those are the frequencies that give us the most resolution or detail information. We'll see examples where it will make sense. And so what we do is we take a normal microscope that has that angled illumination, and then we place that spectral filter that we saw before into something called the Fourier space. So this is the space in the microscope where the light coming from the sample is effectively collimated. And it has some special properties where if you put something in this plane, then you can actually modulate the spatial frequency component of your object. And then we capture multiple measurements using different LEDs, and we do that one at a time. So let's look at what happens when we place the spectral filter in the Fourier plane. Now, if we shine light that's on axis, the spectral filter is in the middle of our Fourier plane. And when we shine light that's off axis, this moves. Now, to get a high resolution picture, we basically need to stamp this all the way around and get information at every single spatial frequency at every single color. And that's our goal. So we take a bunch of measurements using this. And our effective resolution is basically how far out we can go in Fourier space. And so similar to regular Fourier tychography, it's going to be 1 over the objectives NA plus the illumination NA. Now, I won't go into the details regarding this, but just like before, we need to build a forward model or some kind of equation that measures what's happening to our light. So in our case, what we're doing is we're starting with some kind of object, and then it passes through this imaging system, and then we get some kind of estimate of what our measurement is. Then we take our estimated measurement and we subtract it from our actual measurement, just like we did before. And then we take that loss and we back propagate, which means we use it to update what is our estimate of our object. Same as before, we also incorporate some priors because, again, we're taking that 3D data cube and squashing it into a bunch of 2D measurements now. And the hope is we can reduce those number of measurements by incorporating priors. And we solve this reconstruction algorithm. I can go into the details in the reception/Q&A, and we get back our samples transmittance. So this work was all in simulation so far. And that's what I'm going to show. So what we did is we simulated, let's say, a resolution target that has 8 different spectral channels. And so each channel, there's something weird going on. And then we have like 50 to 500 LEDs that are shining onto that object. And then we get a bunch of different measurements. So that's the first part. And we take those measurements and then we feed it into our reconstruction algorithm. We pretend we forgot about our object. We don't know what it looks like. And then we solve. So this, for example, is a picture of the video of the reconstruction algorithm running where it's guessing what was that object that created this measurement. Okay, first we did single color results, which means we only had one spectral channel. And the question was, now we have this weird aperture that's a square. What effect does it have? And we saw that when we have too few LEDs, then we don't have good reconstruction. So on the left, what we're seeing is a plot that's measuring Fourier coverage, which means how far out did we go in Fourier space and how much of that total area did we actually measure? And we want more coverage to get better results, but it also takes longer. So we have to find a sweet spot. On the y-axis is multi-scale structural similarity index measure, which basically is a way of measuring image quality. How close is it to our actual result? In the middle is a Fourier coverage plot. So what that's showing is, again, which parts of that Fourier space did we actually capture? And on the right is a reconstruction of our actual object. So as we increase our coverage, which is expected, our reconstruction quality goes up. Like this picture matches our ground truth and we're able to resolve these bars. So that's expected. And this happens around 1.5x Fourier coverage. And that matches the old situation where we had circle apertures. So now we use this to do multicolor reconstructions. And we have 160 LEDs, 300, and 500. The reason we have to increase the LED numbers is this interesting effect where as you change wavelength, your Fourier coverage changes. So happy to talk about that in Q&A also. But it basically means is for longer wavelengths to get the same amount of Fourier coverage, you have to go out to like 500 LEDs versus shorter wavelengths. And we see that the reconstruction quality is improving with more measurements. But we don't want to spend so long taking measurements. We want to reduce our time spent on acquiring with a microscope. So we incorporate priors. And what that means is we take our low measurement count image, and then we play with these extra parameters that are in our reconstruction function. So for example, we say that, oh, I know that my spectra is smooth. It's not spiky, it's changing very slowly. Or I say that, oh, my image is mostly smooth. It has mostly flat spots and then some dark spots. And then by incorporating both priors, we were able to get the best result that kind of matches our ground truth. So this is kind of a proof of concept that there is some amount of like compressed sensing we can take advantage of and some spectral scanning, or spatial frequency scanning. And when we look at the actual spectra for this is a comparison. The no prior case, we were also not able to get the right spectra for a given point, but with the priors shown in blue, our spectra also matches the ground truth. Only in some parts. Other parts, it's like still a little bit off, so we still have to debug that. Okay, next step is this was all in simulation. I had a student work with me for a semester, Sanjay, who started off with like building an optical setup. So we took a little bit of measurements. So it was a good promising start in that direction, but there's still a lot of work to be done on this project, and hopefully other people are inspired to take it forward as I graduate. And overall, again, the applications for digital pathology, this is like a very simple technique. Also, shout out to Zach Phillips, who lent us that LED array that's shown in this picture. And the hope is like, we could maybe get this working for simulated objects and then try it on tissue samples. So in summary, I presented a few different projects that were all around this theme of hyperspectral imaging for microscopy. The first project, we did spatial scanning and we used a piece of Scotch tape to spread out the light onto a sensor. In the second project, we built this small attachment that can go onto any benchtop fluorescent microscope. And then in the third project, we played with this new idea of if you just add a simple element into our system, we can turn this into a spectral Fourier tychography system. And the theme behind all of these is like, what are small, easy changes we can make in the hardware and then adapt the computation accordingly to be able to incorporate new features and new capabilities into our system. So I'll take questions now before we get to my favorite part. One or two questions? Yes, Ed.
How much computation power do you need for these kind of
Sure.
boxes?
Yeah, so in the first case, that one is non-iterative, meaning what you do is just a simple matrix inverse. That one can be done on the CPU and it takes like less than a minute. In the second case, we got kind of fancy where we have this iterative algorithm running and you can run it for like 1,000 to 2,000 iterations. And the data cube is rather big because we have like 1,000 by 1,000 pixels and then we have 64 color channels. So because of that, you do need a GPU that's like at least 12 gigs to be able to run that reconstruction. And then it takes about 5 to 10 minutes for the whole algorithm to run.
For a single step.
For a single frame. And then we have to do that for multiple frames to do the snapshot. So there is some work where we can improve that using like space-time methods, using redundancy from one frame to the next. And then also if we have a data set, then we could also train a model that can do that inference more quickly. And then the third case, I think that also took around 10 minutes. And it was done on this, that was in simulation, so much smaller. And I did it on a GPU that was less than 12 gigs. Yes. Yeah, that's a good question. So the question is for the third project, can we change the filter so it actually becomes basically a circle to try to fill in the rest of the pupil? I guess is my, yeah, I think that would be the way, is like you make it bigger. Right now we had the circle and then the square was inscribed in it. You could make it bigger, so then the circle is inscribed in the square would be an easy way. We'd have to see how to manufacture a filter array. That's kind of the hard part. I did talk to some vendors and they're able to like mosaic things together, but that's kind of the hard part. Yes, Eric.
You make method where you
Not right now. Yeah, that's another thing we could do. So the question is, can we have white space outside of the filter where you still have Fourier space and that's letting all the light through? So maybe that could be great for getting your spatial prior or like your spatial information reconstructed really well. So that's, I don't know. So like maybe it's useful for the spatial reconstruction and then you have other parts that do the spectral part. Yes, Tan. The question was about resolution and I don't know how to answer it right now, so maybe we can talk about it offline. But I think the answer is like, yeah, whenever you have regularization, it affects where you end up, and that could affect your resolution, because you have to be kind of careful about tuning priors. Any questions on Zoom? Okay, now we're going to get to my favorite part. Okay, so whenever I read a PhD thesis, the only part I actually fully read is the acknowledgements page or pages. And then I skim through the rest of the thesis where I don't read it all. So I was really excited for when I would get to graduate and I get to put this section together. So first of all, thank you to my incredible and amazing advisor, Laura. This PhD was like a long time in the coming. Laura and I met in like 2018. And then I was like, I'd love to come work with you, but I'm also burnt out. Please let me take a year off. And she said, yes. So then I joined in 2019 and it's been such a joy to work with Laura. I will say, like, the things that I've learned from Laura the most is first is how she empowers other people and encourages them to chart their own path. I think we're lucky in our group that we have so much flexibility and freedom to pursue our research ideas and go in any direction. She doesn't demand anything. She doesn't like narrowband or pigeonhole you in anything. So you really have a lot of freedom. And I've spent time reading like organic chemistry textbooks to like machine learning textbooks to like information theory textbooks during my PhD. Now I'm learning like cello and flying. So kind of everything. And then she also supports your vision. So if you do go in a direction and you need resources and support, she helped find several of our collaborators. When you get stuck, she helps unblock and helps you see the problem from a different perspective. And she really uses her network to help you. And the last quality that I really admire is that she draws really kind and really brilliant people to her group. And I've really been lucky to work with all of the members in our lab group. So thank you, Laura, for having me as a student and just for putting up with my weirdness. Yeah, my first group meeting, as I did an intro talk, which of course was a theatrical performance because that's what I do. And she was, I don't think so. Yeah, I don't think anybody was there. It was a time, so ask the old alums. Next is I'd like to thank my quals and thesis committee. So I was really lucky to actually take classes with each of these professors. I took computational color with Ren Ing, who served on my quals committee. I took modern microscopy with Gokul Upadhyayula, who's in molecular and cell biology. And then I took ham radio with Mickey Lustig, who was again supporting my theatrical endeavors. And we put on a radio play during COVID. So that was really fun. Yeah, thanks for serving on my quals committee and just acting as support and sounding board. I really appreciate it. And now we get to read my thesis. A lot of collaborators, I won't have time to name them all, but for the spectral diffusers, that was a second project. There were a lot of people involved. We had people making those diffusers or phase masks. We had people prepping the samples. We had a lot of help between me and Eric on like thinking through the whole concept in general. So really thanks to all of our collaborators and our funding sources. And then for the diffuser spec team, I mentioned we were collaborating with Vanderbilt, and then the Fourier tychography team was mostly me and Laura, and then a few others who were like willing to donate their time. So thank you, collaborators. Okay, I think the best place to be or to PhD is the Waller Lab. I've really had so much fun during my 6 years here. It's kind of why so many people in our lab draw our PhD out to 6 years, because like I have the best job. I get to just think and do the things I like and get paid. So really thank you. I won't have time to name everybody right now, but please know I've learned a lot from each of you in the lab. And we've also just had a lot of fun and weirdness and a lot of adventures together. So thank you for showing up every day, and thank you for doing the science that you do. And then all the other labs. So before I was in the Waller lab, I worked in high school at Rice with Alvin Orbick, who was like my first research mentor, and we published our first paper together. I was in Rajiv Ram's physical optics lab at MIT during my undergrad and my master's, where I learned a lot about how to do research and also how to make research impactful for society. And then I also did a short stint at the media lab when I was wandering during undergrad, trying to figure out what I wanted to do. So thank you, all of the other lab mentors. Also, thank you to all my amazing teachers. I feel like this moment now is a long time in coming. I've been doing science since I was like 7 years old, and I've had a lot of teachers that have encouraged my interest in science. For example, a lot of my high school teachers and a lot of my college teachers, both in the EE and the theater arts department, that really motivated me to go on stage and like have the biggest experiment possible. So that was really empowering. And then at UC Berkeley, I had the privilege of teaching with Steve Connolly and Babak Ayazafar, and I got to, spread my love of signals and systems to the Berkeley undergrads. And then my family. So I'm so lucky. So huge shout out to my parents and my siblings who've been there for me. This is a picture on the bottom right where like my parents and my siblings have been coming to my science fair projects and presentations since I was a kid, and they've always been really supportive. My sister taught me like math when I was 5, 6 years old, and fostering into my love of computation. My brother and I love talking about things like science to international affairs. And then my brother-in-law, Ari, has also just been so kind and so supportive. My mom studied music, and so I'm inspired by her to do creative things. And then my dad really like taught me resilience. They're both entrepreneurs, and so they face a lot of failure over the years, and they've instilled in me the importance of just persevering, and you just get up and you keep trying again. So thank you. And then I have a lot of family because I'm Indian, so bear with me. a lot of extended family in the top left. That's like my dad's side. And then on the top right is all my Bay Area extended family. And then my cousins kind of shown along the bottom. So thank you, everyone. It's really nice to see some of you join today on Zoom. I really appreciate your support, that you took the time to be here. And also just like, thanks for listening to me talk about my weird science dreams. And then my partner, Alexander, who I had the luck of meeting during my PhD. So best discovery that ever happened during my science career. We've done a lot of science together and art and adventures. And I look forward to continuing doing that with you for the rest of our lives. And also my friends and community. So my best friend Vaish is also here today. Thanks for coming. And my roommate Meg joined on Zoom. She lived with me all 5 years during our PhD. Kramnik and I started our PhD. Daniel Kramnik and I started our PhD to get not at the same time. He started before me, but he was kind of a constant. And then Clarissa was also my friend. Oh, before we leave, let's do a really quick group picture. I know I have one more. Bye, Alexander. Wait.
I'm late for meeting with my boss.
I know. Take a quick picture though. Okay, he's leaving. In case anybody else needs to leave. Okay. All right, and then more friends. This is the last slide. Thank you so much, Ruth and Michael, Ned's parents, and Ned have their home away from home in Compchi, and yeah, just so many other friends. I know people have to leave. Right before you leave, if you want to turn on your camera, then we can do a Zoom screenshot. Then I'll let you go. So we did the in-person selfie. I meant to do this earlier. If you turn on your camera on Zoom, I'd love to take a photo of everyone that's joined. Oh, thanks so much. Yeah, and it's great that just people from different stages of my life are here today, and that it means a lot. So thank you, and hopefully our paths intersect again. Okay, I'm gonna take a photo. Okay. Great. Well, this is the last slide. So thanks, friends. Thanks, everyone who's here. We have a reception. Oh, we have one very important question.
Grad student or like a more junior person, and who was a business go down forever etched into.
Yeah. So Laura asked her seminal question, which is, what are my words of wisdom for a new grad student? And we keep track of all of this in our lab. So I prepared very carefully for this. And I will say my words of wisdom are, remember that the most important creation of a PhD is the student. And this is your first original scientific work, not necessarily your like last or best or greatest. So really take the time to invest in your own personal growth. And remember, you are the best output of your PhD. And then I have more advice, but you can ask me offline. Well, it's us, it's you, and then how can you grow and support others? Yeah, it's good. All right, thank you everyone for coming. Really appreciate it. Thanks, friends. Thanks, cousins. Thank you. Thanks, both. Thanks, Mickey. You said what time? What time's our food? What time's our food? Okay. Cool. Yeah, we can show. We have Girl Scout cupcakes and we have drinks in the bags over there. Thanks for coming, everyone. Oh, Mom, Dad, I missed you guys in the photo. Let me take another one. How do I switch to gallery view? Oh, okay.
Neerja, congratulations, Mosaji and me.
Thank
Congratulations,
you.
Neerja.
Thank you.
Congratulations.
Let me take a photo. Is Mom there? Does she want to turn on her? Mom, do you want to turn on your camera?
Oh no, Mom inside. She's not listening.
Okay.
Like she only listening, but there were a lot of crowd, so we are at the park
Sure.
outside the wedding.
Mary, thanks for coming.
Thank you, Bete. God bless you. God bless. See you soon.